Hello, I'm Romanos Sklavenitis-Pistofidis from the Dana-Farber Cancer Institute, and in this meeting I'm presenting results from our single-cell RNA sequencing cohort of bone marrow and blood immune cells from patients with high-risk smoldering multiple myeloma. So smoldering myeloma is a very heterogeneous disease. We have patients who may never progress and patients who will progress really quickly, and therefore it is crucial to understand who is going to progress and then potentially treat them early and prevent progression with what that brings, and organ damage like kidney damage and bone disease. So this far we have good clinical models that do exactly that, and we have a slew of papers that look at genomic biomarkers. So can we look at the mutations that are inside the tumor cells and be better at predicting who is going to progress. What we're doing here is we're bringing immune cells in the equation. We're trying to really understand how we can leverage the patient's immune state to improve our predictions of progression to myeloma, response to therapy, to understand what therapy to give to what patient. So we use single-cell RNA sequencing. There is a really nice analogy people use to describe it, where we go from the smoothie to the fruit salad. When we use bulk sequencing approaches, we extract RNA from all cells in a single sample, and of course within that we may have tumor cells, we may have T cells, we may have B lymphocytes, monocytes, etc. And we can, when we look at the data, we cannot really tell what piece of the RNA comes from what type of cell. With single-cell RNA sequencing, we can map it back to the cell type, and now with a single sequencing run, we can profile both the tumor cells and the T cells and the B cells and so on and so forth. So in our data set, we had 190 patient samples, which is to date the largest data set, and it allowed us to really profile all of the different changes in immune cells that we see in patients. So we saw increases in certain T cell subpopulations and decreases in others, and really the challenge here is understanding how to leverage that for prognostication. Sure, we see the changes, but what do they mean for patients? And so what we saw was that patients who are the least normal are actually the ones that have the longest progression-free survival, which was really interesting. And not only that, but also patients increase in normalization post-therapy. There is this idea of immune reconstitution that's been going around in the myeloma field for many, many years now, and now we actually are able to see that patients are able to normalize their immune system with therapy, and we see that that normalization, post-therapy immune normalization, or PIN, may actually be better for them. They have longer progression-free survival. And perhaps the most exciting finding of our study is that before we needed to do a bone marrow biopsy in order to profile the immune state of the patient, now we see that pretty much the same immune dysregulation we observe in the bone marrow, we can actually also detect in the blood. So imagine a scenario where we can now just do simple blood draws regularly. You know, nobody minds a blood draw as much as they mind a bone marrow biopsy, and profile their immune state and include that into all of those risk stratification models that we use. We think that this is really the way to the future. We envision a future with both clinical parameters, genomic parameters, and immune parameters, helping to improve our prognostication for our patients. In older years, if you wanted to do the exact same thing, you would have to do a bone marrow biopsy, isolate immune cells. You don't know what is what, you just have a sample of cells. And then you would have to do maybe flow cytometry or some other method that allows you to sort individual cell types and sort each cell type separately, extract RNA, and sequence each of them separately. But in our data, we have 63 cell types. You cannot do that. It's just very hard to do without single-cell sequencing. Yeah, so this is the beauty of single-cell RNA sequencing. You don't necessarily have to pick the cell type you want to look at. You can just take the sample, sequence all of it, and then you're going to be able to deconvolve it into all of the cells that were in that sample. So in a typical bone marrow biopsy single-cell RNA sequencing run, we will detect tumor cells, we will detect normal plasma cells depending on the stage of the patient. We will detect T lymphocytes and B lymphocytes and monocytes and dendritic cells, essentially almost all of the immune cell types that we know today. The trick is, how do we go past that detection? Sure, I know that you have more of this particular subpopulation of T cells, but what does it mean for you? Is it a good thing or a bad thing? And I think that is really the challenge and the frontier with these studies, and that's what I'm hoping we're going to get to hear more about in the future years. It means you barcode all cells in a single sample, and then you extract RNA, which is now barcoded. So once you sequence the soup or the smoothie, you're now able to tell that this piece in the smoothie came from a strawberry and this piece in the smoothie came from a banana. So you do sequence single cells. That's why it is called single-cell sequencing. But for sequencing to happen, you actually have to break the cells and make a smoothie. That's what's actually sequenceable. That does not necessarily give you spatial information. You don't know where in the bone marrow these cells were and how they were arranged in space, but there are newer technologies that are coming about that actually enable you to do that. But for these technologies to work, you have to start off of a biopsy slide. You have to have some sort of spatial information for the cells before you barcode them, and then you have, let's say, extra barcodes that enable you to map these cells in space. This is very important to us as well, but it is the next frontier. There is an added difficulty in myeloma, and the reason is because myeloma is in the bone. In order for us to make those paraffin-embedded blocks from which we cut slides and then we do all of the spatial transcriptomics that you're referring to, we have to decalcify the bone. Otherwise, we can't cut it. It's too hard. And by decalcifying it, unfortunately, we are destroying a lot of the DNA and the RNA that is in there, and that, obviously, is a problem we cannot work around. You have to be, wise about it and use a solution that preserves nucleic acids in order to be able in the future to do that.